Okay, welcome everybody to this month's JSPS Pathology Grand Rounds, as sponsored by the Shanks Foundation and the Pathological Society. Can I remind the audience, as usual, if you have questions for today's speaker, please can you type your questions into the Q&A box at the bottom of your screens, and we will have a question and answer session at the end of the lecture. Now, it gives me great pleasure to introduce today's speaker, who is Dr. Tjalling Bosse. He's currently an associate professor in the Department of Pathology at the Leiden University Medical Centre in the Netherlands, where he combines clinical work, teaching and research. He received his MD degree in 2008 from the University of Amsterdam and his PhD degree from the Erasmus University in Rotterdam. He completed his pathology training at Leiden and specialised in gynecological pathology, and that included doing a fellowship at the Brigham and Women's Hospital in Boston. Currently, he leads a translational research programme focused on gynecological cancers, and he's the lead pathologist for some international randomised controlled clinical trials, including the PORTEC and RAINBO-trials. His research currently has 3 active themes: improving diagnostic accuracy by integrating molecular genetic features in endometrial carcinoma, identification of novel druggable targets within endometrial carcinoma, and utilising deep learning to improve diagnostic accuracy and identify novel prognostic and predictive biomarkers. And he's going to talk about some of these aspects in his lecture this morning, which is entitled The Molecular Classification of Carcinoma. Thank you very much indeed for agreeing to speak to us this morning. Over to you, Tjalling.
Thank you for the kind introduction. I'm gonna start sharing my screen for my presentation. All right, good morning, everybody. We have, I understand, 1 hour, which is going to be very full and data rich. I have included 100 slides, so I will entertain you quite a bit. Hopefully, there will be sufficient time for questions. I'm looking forward to it. And as mentioned, today's talk will be about endometrial carcinoma and in particular the molecular classification. And it's very timely to talk about this particular topic, both from the angle of the fact that there's been a lot of change in the last decade or so, but also because endometrial carcinoma is on the rise. And in particularly, this is, of course, seen in all the statistics. And since I'm talking to a UK audience, I looked up the Cancer Research UK incidence rates in for endometrial carcinoma, and we can see that there is a clear rise in incidence in the UK. And this is not unique for the UK. In fact, there's a global rise, especially in the high and high middle income countries, which was nicely shown in a recent review by Emma Crosby from Manchester in the Lancet of 2020. And what was actually quite interesting in this particular review is that there was highlighted that when they looked at the most marked increase, in fact, that these are found in the low and middle income countries. And the observation links probably to the obesity pandemic that is going on in combination with an increase in age also in these countries. Well, what does this mean? This means that there's a major healthcare burden coming at us, and we are going to have a lot of work to do, a lot of work for pathologists, but of course for the complete management team. And this is also an interesting article where they try to project the rising incidence rate into the future. And this is from the United States, where it is projected that uterine cancer incidence will surpass colorectal cancer by 2030. So it's really an important cancer subtype to study and to work on. we know that it often, it generally occurs postmenopausal and with symptoms of postmenopausal blood loss. And then usually it's a low stage situation where the tumor is confined to the uterus. Primary treatment, cornerstone treatment would be surgery. This is pretty well worked out, but then the question about adjuvant treatment has been under study for many decades now, is how to best align the adjuvant treatment in this scenario. Should we not give adjuvant treatment, give radiation, chemo, a combination of radiation and chemo, and or should maybe novel targeted treatments already move towards the adjuvant adjuvant setting? And working this further out will be very beneficial for this enormous rise that is coming at us because of the, of course, economic burden of these adjuvant treatments. For a long time, this adjuvant treatment has been based on a certain risk assessment in which pathology actually has a central role. So generally in tumor boards, pathologists have a really important role in the management decision with regard to adjuvant treatment for endometrial cancer patients. And this is because a risk an individual risk is developed based on several variables, mainly clinical pathological variables, such as stage, histological subtype, grade, and the presence and extent of lymphovascular space invasion. This risk assessment, however, comes with several problems, mainly, and we know this now for quite some time, and it's pretty extensive. There's a high inter-observer variability, particularly for the pathology variables, of course, for all of them, in fact. Stage, so is it stage 2, yes or no? Is it stage 1B or 1A? A lot of inter-observer variability there. Histological subtype, particularly in the high-grade tumors, and LVSI is also challenging. So this is causing actually a system that is not very robust for treatment decisions. And then in addition, this lacks actually a biological basis which would be needed in order to move towards, for example, more targeted treatments. So we've known this problem for some time, and it's actually happening in many other tumor sites, of course. And one solution that people think of, and we also thought of, is maybe we have to move towards molecular classification. And this is exactly what has happened between the WHO of 2016 and the current 2020 WHO. We moved from a histologically driven endometrial cancer classification, where we classified tumors based on whether they were endometrioid, looking like endometrium, or non-endometrioid, and then we had several subtypes, and where we used diagnostic immunohistochemical markers upon indication when we felt it was needed, towards a novel classification system, which has a very clear biological basis and divides endometrial cancer into 4 subtypes based on a important molecular driver. And this is all started back in 2013, where the Nature paper from the Cancer Genome Atlas came out and first set out this concept of 4 molecular subtypes. And here I show you the first figure of the Nature paper, very data rich, in which there's a lot of information about a large set of endometrial cancers. I think it was close to 300. And importantly, one thing that I always, when I look at this, see is that every endometrial cancer is very different. And they were able to identify 4 sort of groups by clustering these tumors based on tumor mutational burden and copy number status. And what you then get is 4 groups. On the one hand, you have the ultra-mutated tumors that are driven by mutations in the exonuclease domain of DNA polymerase epsilon. That was kind of a novel finding at the time. Then there is about 30% microsatellite unstable. On the far right-hand side, there are the tumors that are copy number high and are mostly driven by mutations in TP53. And then there is a group that the TCGA called copy number low, which are more or less the stable tumors that tend to be of low grade and in endometrioid histotype. Very quickly thereafter, we figured out, and with us some other groups in the world, that this, of course, can also be replicated by using very easily and pragmatic surrogate markers using immunohistochemistry and targeted sequencing, so that you don't need the whole exome sequencing or whole genome sequencing, such as performed by the TCGA. The TCGA wasn't really set up for prognostic assessment. There was not a lot of clinical data, but the interesting observation they did is that these groups are also prognostically different. And one, of course, particular interesting finding was that the 17 POLE mutant endometrial cancers in the TCGA didn't show any progression and any recurrence, so flatline. So very quickly thereafter, we moved towards looking in large data sets, in this particular study at 2 trials, PORTEC 1 and PORTEC 2, which contains low and intermediate risk endometrial cancers, low stage, that were 2 randomized controlled trials. And we performed the molecular classification and found strikingly similar findings, particularly of interest, of course, the flat line of the POLE mutant cancers. And this time there was a lot more cases and robust long-term follow-up. Subsequently, we started to look into more specific subgroups, such as an international collaboration with grade 3 only endometrial carcinomas, that we published. And very interestingly, up till that moment, people were treating grade 3 endometrioids as one disease entity, but as you can see from this graph, the Kaplan-Meier curve very clearly indicates that this entity is actually not one entity, but there are 4 groups. And this is already back in 2018 and has been replicated several times. And also a very active group in Vancouver has done similar work, particularly the group of Blake Gilks, and they showed in unselected non-trial cohorts that these Kaplan-Meier curves are reproducible. And I could probably show another 10, if not 20 papers that show exactly the same profiles. So really convincingly, the molecular classification carries prognostic information for endometrial carcinoma. And one of the more recent studies that we did was on the clinical trial called PORTEC 3, which includes only high-risk patients that were considered for the addition of chemotherapy. And also in this trial, the molecular classification showed prognostic stratification. And again, the POLE mutant group of tumors, which was about 10% of all, showed a flat line in this cohort. A question sometimes is asked with regard to the P53, because not in all these trials patients have been fully staged by lymphadenectomy, is whether they are missed high stage cases. This was resolved by one of my PhDs, Alicia Lyon, who worked together with a Danish group where they do full lymphadenectomies on endometrial carcinomas. And we could show that the poor P53 outcome is also found in stage 1 cases that got a full lymphadenectomy. So there's no hidden high stages in there. Then a question I often get is, what about non-endometrioid? So serous and endometrioid is clearly working for, but the more rare entities, such as uterine carcinosarcomas, clear cell, and D-diff carcinomas, is more difficult and challenging to study. But what we do see in the literature is small cohorts being published in which, and at least we can say that the POLE mutant endometrial POLE mutations occur in all these also more rare entities. And for the uterine carcinosarcomas at the most recent USCAP, Brooke Howitt from Stanford showed a cohort of, I think, 150 cases, which again showed very nice stratification. So definitely also uterine carcinosarcomas can benefit from this. And just as one step outside the uterus, also endometrioid type ovarian cancer has been studied. This was worked together with, it was a big international collaboration close to 500 cases. This is quite a lot for a rare entity like this. And again, very strikingly, a flat line for POL-E. Okay, now we have seen all this. There's a certain hierarchy to this diagnostic algorithm. This was how the TCGA had put it, but this is how we envisioned it or have proposed it for from a surrogate marker approach. So it starts with testing POL-E, subsequently MMR status, and then it ends with testing for P53 status. And this diagnostic algorithm was not just made up as, this was based on actual data, and goes back to the story we did on what we call the multiple classifiers, because mutators like microsatellite unstable or POL-E mutant cancers, they can of course also have P53 mutations. So then you have something good and something bad in one tumor. And the question of course is what drives this? And what is the clinical behavior of these, what we called multiple classifiers. This was published in Journal of Pathology by this group of excellent collaborators. It was a really fun study to do, very international, because it was very challenging to get all these cases together. And Alicia Leon, former PhD of mine, is first author on this work. So she found these multiple classifiers in all these cohorts through international collaboration. And as you can see, it's a very small subset that has this problem, let's say. And what is it that she found is if you look at cancers that are MMR deficient and show a P53 abnormality by immunohistochemistry or by sequencing, is that if you look at the immunohistochemical pattern, actually quite a significant proportion, 40%, shows diffuse P53 mutant pattern. However, 60% showed something we call subclonal mutant expression, where there's an area, geographical area mutant, but another area completely wild type. This already somewhat suggests that it might be a secondary effect of secondary mutation in the context of such a mutator. And this was further corroborated by hierarchical clustering we did on the TCGA data, where in red you see the single P53 mutant cancers, in green the single MMRDs, and then in brown the combined multiple classifier cases. And the brown seemed to cluster with the green, so from a copy number status, it was clear that they are clustering with single MMRDs. And then of course the most important finding was the clinical outcome, where the double classifier MMRD P53 were actually showing quite good clinical outcome. And if we put that next to a reference group of single P53, it was clearly much better. So this really suggested that MMRD P53 combination should be regarded as an MMR deficient endometrial cancer in the diagnostic algorithm. And this picture looks very similar, and that's because it is very similar. In the POL-E mutant combination P53, we see basically the same thing happening with lots of subclonality, clustering of the double with the single POL-E, and also with outcomes that look like a POL-E mutant cancer. So having a secondary P53 mutation in the context of a mutator doesn't seem to drive the tumor. So that means all these data have led to the fact that it's now incorporated in the WHO with a diagnostic algorithm as shown, and also clinical management decisions are being associated with it, as it came from this guideline paper from the combined ESCO, ESTRO, and ESP. So that means we have to start testing. And then, of course, we have some issues to talk about, because if you start to test cancers for POL-E, you will also find variants that are actually do not qualify as a true pathogenic POL-E mutation. And this is particularly in the context of microsatellite unstable tumors where you find these. And this causes somewhat of a challenge. So we worked this out, and importantly, POL-E mutations that we're really looking for are obviously pathogenic. They are inside the exonuclease domain, and they are associated with signature 10, which is very different from the microsatellite unstable signature, and of course associated with a really high tumor mutational burden. And this was also work by Alicia, who looked at all the publicly available data at that time, and looked at which Hotspot mutations are recurrent, which show this signature and the poly signatures really don't have any indels. They have a propensity to have C2A and T2G substitutions. And this has resulted in a list of accepted pathogenic variants, a list of 11 hotspots that qualify as being a polymutant endometrial cancer. And really the first top 5, let's say, are the ones you will find in your cancers. The other ones are very rare. But it is very important to be aware that you can find polyvariants other than these 11, and that this doesn't necessarily qualify as a polymutant endometrial cancer. Of course, if you have 11 hotspots, you could consider not doing an NGS panel, but more targeted approaches. And a lot of companies have started to develop test kits. We also developed an academic, very simple qPCR methodology, which reduces the price a lot and the turnaround time also a lot. So this could be an interesting step when we think about making it affordable for all countries. Of course, we're pathologists, so the first question you ask is, how do they look? At least that's what the question we asked back in, 2017-18. So we did a blinded study. This is me when I was still young in my other office. And we collected 150 cases, of which 50 were polymutant, and the other 100 were not. And I was not aware of that, and I scored about 50 pathological variables. And what was very striking in the first observations back in the day, now we all know it, is that these cases, these polymutant cancers, they are really filled with lymphocytes, both intratumoral as well as peritumoral. And there's a lot of tertiary lymphoid structures identified in the myometrium, which resulted in the study where we also found one other feature, which is that the polymutant cancers carry these crazy scattered tumor giant cells. Was another observation we did, and often they are of high grade, which is why we called the paper sheep in wolf clothing. And later we found these associations with certain immunohistochemical patterns, such as subclonality of P53 and MRD. Can you identify them on an H&E? Maybe you can, but it's not sensitive enough. So probably sequencing will still be necessary. Then we worked on another project together with the UK. This is David Church, medical oncologist at Oxford, and Inge van Gol, another very bright PhD from my group, who took on the challenge to figure out how this all works. And we got this nicely on the cover of CCR, which we really liked. So she went a little bit deeper into the topic and found indeed that these lymphocytes are active, are T cells, and that they are CD8 positive T cells, and that you can find them intraepithelial, but also in the surrounding stroma. And we showed that these lymphocytes are also really active with granzyme positivity and TIA1 positivity in these immunofluorescence images. We also looked at the TCGA data, of course, and found an interesting observation that there's also quite striking upregulation of immune suppressive molecules. Particularly interesting, of course, is PD-L1. And Brooke Howitt in Stanford showed also on a protein level that there is an increase in PD-L1 expression in the polymutant cancers, which of course has led people to look at the possibility of using checkpoint inhibition for the treatment of these patients. And there were a couple of case reports published at the time showing good responses indeed in polymutant cancers with metastasis, which is very, very rare. But it clearly showed an effect which made total sense at the time. Of course, the majority, however, will be early stage. And if we look, for example, at the PORTEC 3 with more than 50 polymutant endometrial cancers, where there were 2 treatment arms, chemo plus radiation and radiation alone, because at the time we didn't know whether the addition of chemotherapy would be of benefit in high risk endometrial cancers. So these are cancers with bad features, grade 3, non-endometrioid, LVSI, stage 2. But as you can see, the polys are showing a flat line. So really giving them chemotherapy is probably a really bad idea. And we even went a little bit further because, of course, if the clinical outcome is every time so good, are they super highly, because many of these cohorts, these patients have been given adjuvant radiation therapy. So one hypothesis was possibly these are hypersensitive to radiation therapy. So we created, Inge van Gol did, created polymutant embryonic stem cell lines, and we started to radiate these cell lines and looked at the possibility whether these are super sensitive to radiation. And what this survival graph shows is, in fact, that there is no, not at all such a thing. The orange line shows a radiosensitive cell line control, and the other lines are different polymutants. And you see there is no difference between the wild type cell lines. So there was no indication in the lab that this poly mutation causes hypersensitivity to radiation. And then we went back to an old trial, PORTEC 1. Why? Because it had a no additional treatment arm, so no adjuvant treatment was given. And then when we look only at polymutants in that no additional treatment arm, we see a flat line again. Possibly this means that these patients simply have so the indolent behavior is due to the tumor itself and not the treatment. And this was again validated basically in the Danish cohort, where in Denmark they tend to give no adjuvant treatment even in grade 3 tumors. And here this is a larger cohort showing again a flat line for polymutant cancers. So this is the first RAINBO study I'm going to share with you, which is the polymutant blue trial led by Canada. And it's currently open in Canada, and international activation also in the UK will follow in 2023, where polymutant cancers treatment will be de-escalated. So no adjuvant treatment, and there will be, so this will be the first prospective assessment of this finding that we've done over and over again retrospectively. Okay, let's move to MMRD or MMSI tumors, mostly endometrioid, often grade 3, also mutators. And also here we find upregulation of immune suppressive molecules. And what is of course important to note is that 10% of these patients have Lynch syndrome, which I always like to cite my friend Robert Soslow at the MSK, who when he presents this always says this is the pathologist's chance to save lives. It's very important to identify patients with Lynch syndrome, and we of course all aware of this. We screen by doing MMR IHC, and this is relevant for the patient for potential secondary cancer that can occur, for example, colorectal cancer, but of course also for family members. And if we look at our large cohort, indeed about 30% is MMR deficient. And if you look on the total population, we see 3% having Lynch syndrome. But within those that are MMR deficient by IHC, 10% has Lynch syndrome. And if we look at the clinical outcome, does it actually matter for clinical outcome? That is rather subtle. It's not statistically different in our cohort, but we do seem to see that those that are sporadic due to methylation seem to do a little bit worse. A few notes about immunohistochemistry. Of course, you can have the classic loss of MLH1p-2 or MSH2 and MSH6 that go together. But you can also have triple negative MMR staining, which we usually see. What the most common combination would be is loss of MLH1p-2 and secondarily subclonal loss of MSH6. This is due to a microsatellite unstable tract in one of the exons of MSH6. And of course, you can have single loss of either MSH6 or p-2. The inter-observer agreement in many papers is very good. So this is an excellent approach. And also there is a very high concordance with microsatellite instability. It's not 100% because you're, of course, testing two different methodologies. Another thing to note is that MMR deficiency can occur during tumor progression, which also means that you can have subclonality of, for example, the combination of MLH1 and p-2, where you can identify promoter hypermethylation in the majority of cases, but then you also have an MMR proficient clone in the background. And another observation that is maybe fun for the next time you do the stains, also look at the normal glands, because you can identify these things that I call MMR deficient gland foci. And when you see that, it's very likely that this patient has a Lynch syndrome, because this is what we exclusively seem to see in Lynch syndrome patients. Testing is done usually by performing all four, but it is also possible to do a so-called 2-antibody approach, where you start with PMS2 and MSH6. If they're proficient, you're done. If you're deficient, you continue with the others. We did a meta-analysis, and this 2-antibody approach is super sensitive. And so basically, in good hands, it's safe to perform a 2-antibody approach if you want to save on some costs. Now, how shall we treat these patients in the adjuvant setting? In PORTEC 3, again, remind you, chemotherapy plus radiation and radiation alone. We actually don't see an effect of chemotherapy. So giving them chemotherapy, probably not a good idea. But as we now know, and very recent data are very strong in that sense, is that certainly in the advanced and recurrent setting, we see very striking and positive results with checkpoint inhibition for this group of tumors, which is, of course, also true in a lot of other cancer subsets. These are just a list of trials that all show very positive results. What is very exciting, of course, within the context of RAINBO, we're trying to push this now into the adjuvant setting. And since we didn't see an effect of chemo in addition to radiation, MMRD Green trial, currently open in the Netherlands, will be opening in the UK, hopefully this year. We will be recruiting MMR deficient endometrial cancer patients, and they will be getting checkpoint inhibition on top of their radiation. Okay, moving on to copy number low. We started calling these no specific molecular profile. I also tend to sometimes call them the triple negative endometrial cancer, because they're MMR proficient, poly wild type, and P53 wild type. And what do we find in these tumors? Well, a lot of Wnt signaling activation and PORTEC kinase activation. A recent study by Lisa Vermeij, one of my PhDs, looked at the molecular landscape of high risk and NSMP endometrial cancer. So this is including many cases that were, for example, at high stage, not necessarily high grade. And what she found is that there's a subset that is ER positive. The majority is ER positive. They tend to be low grade and have a very often mutations in PORTEC kinase and Wnt signaling, very little L1-CAM expression. But then there is also this group of tumors, which is only about 10% of the total, that are ER negative NSMPs. And they have a slightly different profile. What we see basically is that you can therefore divide NSMP into the ER positives and the ER negatives. And the ER negatives tend to have clear cell carcinomas and mesonephric-like carcinomas in them. And not infrequently, they have substantial lymphovascular space invasion. And does this matter? Yes, it does matter. If we look at the clinical outcome, fairly large cohort, you see that the ER negatives are showing actually very bad outcome, comparable to the P53 mutant group, which is why we sometimes see that NSMPs are not such a good tumor. So it really matters your ER status. And Lisa suggested possibly to adjust the algorithm a little bit further and maybe add ER to this. And this needs to be further validated. Two very recent studies already did this, and in fact, they all seem to come to the same conclusion. So indeed, the role of ER is back at the table of endometrial cancer within the context of NSMP. So how are we going to treat NSMPs in the adjuvant setting? Well, here it's a little bit more tricky because there's an unclear benefit for chemotherapy. It wasn't significant in PORTEC 3. Here the question is, of course, chemotherapy gives a lot of toxicity, and knowing that by far the majority is hormone receptor positive, the MMR of the NSMP Orange trial, which will be led by the UK actually, will be performed, where we will compare chemoradiation with radiation plus hormone treatments to see, and this is a non-inferiority design, to see if you get less toxic treatment, but a similar outcome. Ending with the copy number high tumors that are p53 mutants in general. So p53 immunohistochemistry is an excellent surrogate marker for p53 mutational status. Of course, we need to know how it looks. You have the scattered expression in the wild type setting, and then you have the different mutant type expressions, overexpression, classic, can also be subclonal. The null mutant with a nice positive internal control, and probably the least well known is the one where you see overexpression in the cytoplasm. And this always reminds me of the first time I saw this. When I saw this, I'm going to put on my pointer. The first time I saw this, that I went back to my immunolab and said, you made a mistake. But then on repeat, I kept getting the same result. And in fact, this is due to a specific mutation in the nuclear localization domain, of course. So work together with Navina Singh on this, and clearly there's a really good reproducibility for this methodology. Works best on curettings, of course. Well-fixed tissue is necessary, but really you don't need to do sequencing in order to assess the mutational status. And the type of expression also is linked, of course, with the type of mutation. Missense mutations tend to result in mutant overexpression. Nonsense mutations, frame shift mutations in complete absence. And as mentioned, the cytoplasmic overexpression is a result of very specific mutations. Does it matter for the clinical outcome? We asked, so we compared a group of p53 abnormals by mutant overexpression versus those that were having a null mutant or a deletion mutant expression. And as you can see with the overlapping curves, it does not. So in my report, I don't make that distinction except in the, of course, microscopic description. Subclonality is also not infrequent, but I already mentioned this particularly happens in the context of the mutators, MMRD and poly. So if you see this, definitely think of a mutator before you sign it out. This is probably the largest data set for p53 abnormal, almost 240 from the MSK with the nice extensive panel. And there's a lot of information, of course, in such heat maps. But what I find always fascinating in this particular is the bottom here is that you see that only 50% of the p53 mutant cancers are actually called serous. The other 50% is other histological subtypes, including low-grade endometrioid. And we actually see that sometimes, that low-grade endometrioids can be showing mutant overexpression. And this has been reported also in several retrospective studies that you have this counterintuitive combination of low-grade and p53 mutants. And in fact, this was a talk of the recent USCAP by Lisa Vermeij again, with a lot of international collaborators, really great pathologists, among which, of course, UK pathologist Navina Singh. And we did a really fun study because we wanted to look at these low-grade p53 mutant tumors, and we thought, well, are they double classifiers? We have to exclude that, of course, because we want to look in the context of non-mutators, so we excluded those. We got a large cohort of what somebody had called low-grade and happened to be p53 abnormal after molecular classification. And we ended up with a set to work with 55 cases. And basically, we showed it to all these pathologists and asked them to re-review because we thought maybe there has been a mistake. And indeed, there was a subset of 17 that none of the pathologists actually called low-grade. So that's just noise in the data sets that we have. But, and of course, these are like, these images are more like glandular variants of serous carcinoma. However, there were also cases that more than 3 pathologists truly called low-grade. And what was fun about this study, we did not tell them that the immunohistochemical result of p53. So they looked at all these cases and called them low-grade, but in fact, they were p53 mutant. And these are just a few examples. So what's the molecular landscape if we look at these tumors? So this is, of course, a very busy picture, but I think it does give some information. So this, I'm highlighting here the tumors that more than 3 pathologists called truly low-grade, but were p53 mutant. And if we look at the molecular landscape and immunohistochemical findings of these tumors, we see that they are actually having quite high copy number status. If we use shallow whole genome sequencing, we see quite significant instability. At the same time, we see mutations that we know of, the tumors more in the NSMP category. And the immunohistochemical pattern shows ER, PR positivity in the majority, and patchy P16, which actually fits far better in the endometrioid category. So they really, the naming of them as being endometrioid is probably correct. Again, looking at clinical outcome, of course, in the middle are those tumors. In blue, these are the tumors that more than 3 pathologists truly assigned as low-grade endometrioid, but were P53 abnormal. And if we put them against some reference groups, they certainly behave worse than your usual low-grade, let's say, NSMP tumor. So these were the conclusions from that study. So they truly exist, this unusual combination, and it matters. So how to treat the P53? Well, here in the PORTEC 3 was a significant finding. So there's truly an effect of chemotherapy in this group, which seems to also make sense. So we have to continue giving chemotherapy to these patients in the adjuvant setting. Of course, this is still a poor outcome, so we should look at other targets. One option would be HER2 amplification, HER2 targeting. 25% of the amplifications of the P53 show amplification, and it's really restricted to this group, so you don't need to test all the others. But another very obvious candidate would be HRD targeting, of course, in parallel to the ovarian cancer work. And this is exactly what we're doing in the RAINBO P53 RED trial, where for the first time PARP inhibition will be added to the chemotherapy in the adjuvant setting. And this trial will open also this year, led by Alexandra Larry in France. So this looks like a wrap-up. We have a solution, but of course we also have created a new problem. There's costs and turnaround times added to the classic system. It's better, it's more robust, but new problem has arisen, which means we also have to look for a new solution. And that new solution might be the integration of artificial intelligence. And I know that there's been other grant rounds before me talking about artificial intelligence options. And I just wanted to sort of end with a few slides on the work Sarah Vermont from my group has done. So wouldn't it be cool if you could molecularly classify based on the H&E through deep learning? So she built a model that by an input of one H&E image can give you an output of the molecular class. And she did that by training a set of more than 1,000 molecularly classified endometrial cancers, and used an intention-based state-of-the-art deep learning model, and then tested it on an unseen test set, which in fact was the complete PORTEC 3 trial. And what she found is very impressive areas under the curve, particularly P53 works really well, but also the other subsets are giving quite good predictions. And if you run only the image-based predictions and look at the clinical outcome, you see a stratification that looks like the true molecular classification. But of course, there are a few miscalls resulting in, for example, not a full flat line for POLE mutant endometrial cancer. But I'm very confident that when we move to new versions of this, that we can maybe use this as a pre-screening tool. But as a pathologist, more fun is the part where we reverse engineer this black box of artificial intelligence. But because what you can do is, okay, based on, so it's making these predictions, but based on what visual information, and that's exactly what she did here. And what you then get is image gallerias that are showing basically the prototypical POLE mutant, which is solid growth, high density of lymphocytes, and presence of tumor giant cells. We all knew that, but the model had learned it on its own, unsupervised. So that's also a kind of a sanity check that what we are looking at, what we have found is correct. For MMR deficient tumors, these are the typical features. So a lot of solid growth, also lymphocytes. And if we move, we see that the P53 image gallery is totally different, with much more nuclear atypia and rect luminal surfaces and a low density of lymphocytes. For NSMP, again, a very different image galleria with more smooth luminal borders, glands, squamous differentiation, and low density of lymphocytes as well. So really, the deep learning model also teaches us a lot. And one thing I thought was very fascinating are the attention maps. This shows a heat map where the model is looking at for making its prediction. And what is striking to me when I saw it at first is that the model actually also looks quite a lot outside the tumor, in fact, in the myometrium. And when we closely look, we see already an overview. This, of course, is a polymutant cancer because it's full of tertiary lymphoid structures in the myometrium. And it shows that we shouldn't be so tumor-centered sometimes as pathologists. And of course, she also quantified this by using the hovernet on the highly attended tiles. And then you get these nice spider plots where you can see what is the contribution of the different cells to make the prediction. I mentioned the model is sometimes wrong. For example, this is a Kaplan-Meier curve of only p53 mutant copy number high tumors, but the green line shows the cases that the model called MMR deficient. And we know why, because it was seeing inflammatory cells. And so the model is wrong, but if you look at the Kaplan-Meier curve, it may actually not be so wrong, because it actually identified those that do a bit better because there are lymphocytes in there. And I'm going to end here. Of course, the molecular classification alone by deep learning is fun, but more exciting is, of course, the next step. And this is the work Sarah presented at the AACR just a month ago. And here she basically combines the molecular classification with stage information and image information to predict and to train the model to predict distant recurrences. And basically what happens is you as an input one H&E image, and you get an individualized risk score for distant recurrence. And we hope to publish this soon, but what I can tell you is that this outperforms every other risk model that we now have made by human-made variables. So that's a really exciting prospect. Also, when we think again about the enormous burden that is coming at us in the near future. So with that, I want to thank you for listening. And I want to, of course, thank all my national and international collaborators and the patients that participate in all the trials that we work on. And I'm happy to take questions.
Thank you very much indeed, Tjalling. That was a really interesting talk. A lot of data. You covered a lot of ground. Thank you very much. Now, can I remind the audience members, please, to type your questions in the question and answer box at the bottom of your screen, and I will read them out and put them to Tjalling for his view. So I'd like to start with asking the first question, if I may. And I'm interested, I always was concerned that the group 3 might be seen as a bit of a dustbin, where you couldn't fit cases into any of the other 3 categories, so you throw them into the dustbin of group 3. So I was very interested to hear about the progress you've made, particularly with the ER positive and ER negative cases. But my feeling is that that's probably not the whole story. Do you think it's going to be possible to subdivide group 3 even further than that?
There's been several attempts, right? So the copy number low or NSMP, however you want to call them, they are quite large. And some also indeed call them the rest group. And there's been several attempts to further subdivide them. An important observation, I think, is that if you look at generally from a classic standpoint, the low intermediate group, and you do the molecular classification, the NSMPs, they're sort of like having an intermediate clinical outcome. But as soon as you start to include high risk or high grade tumors, the NSMPs actually drop to the level of P53 in terms of outcome. So what the signal we're getting is that actually classic pathology variables are starting to become less and less important in the other groups, such as grade and stage. For example, if you look at grade in MMRD, it doesn't do anything. If you look at histological subtype in P53, it doesn't do anything. But within NSMP, it does. So the relevance of grade, stage, LVSI, still remains very powerful in the context of these NSMP tumors. And ER is just, I mean, the observation is nice because we all have ER, right? And we can do it and we know how to read it. It's pretty easy. A lot of people were already doing it because of the potential future hormonal treatment these patients would be getting. So, and the plain negatives, it's also sort of it's helping you to not miss a mesonephric carcinoma because that's where they are. It groups the clear cells. So indeed, it's at least one step to sort of nail down the real bad ones, and the ER can really be of help there.
Okay, thank you. And just on the same topic, how did your AI deep learning algorithm get on when it was subdividing those group 3 tumours? Was it able to pick out ER positive versus ER negative without doing the ER stain?
Yeah, so very important point. So visually, these ER negatives are probably very different, right? But they're also a very small subset. The study I showed was already enriched for high-risk cancers. If you would do this on a population, you will only find them to be, well, I would say less than 5% of your NSMPs. So the model didn't see a lot of them, and we also didn't train the model on this 5 classification. We trained it on a 4 classification. So we basically haven't refined the model for this subgroup yet. It's a very obvious next step that we could pursue, and we might.
Okay, thank you. So there is a question in the Q&A box. This is from Terence Smith, who asks, did your AI pick out the subset of low-grade endometrial carcinomas that were P53 mutant, and what features did it use?
Very, very detailed question. Very good. You know, I cannot. This is, of course, these are big studies, so this is, and those cases are not so common. I do remember at least one case, but that's then in one case. I don't like that in my answer, but I do at least remember one case where the model called it NSMP, and it was a low-grade P53. And I think the model has learned to appreciate very much so the nuclear atypia. And if we look at the nuclear atypia, the low grades, not for nothing, are called low grade, right? Yep. So at the moment we're struggling, and I think the deep learning model is also struggling because it doesn't seem to have a visual feature. And even though what might be going on is because we also did some ploidy studies, is that they are actually still diploid. So maybe these are very early P53 mutant tumors that if you catch them a little later, they will have more nuclear atypia, let's say. So I suspect they do progress, and that's also what we see in the outcome data. But apparently they don't have the visual features that we know usually is associated with a P53 mutation, which probably is the result of the genomic chaos in these nuclei.
Absolutely. Thank you. So can I take you back to ask you a question about the second group, the MSI group? And you described this feature of where you can get an MLH1 mutant tumor, which sometimes shows subclonal loss of MSH6, and as you explain that with the poly A tract in the coding exon, which is a target for the mismatch repair pathway. The question I want to ask is, do those cases where you do have MSH6 subclonal mutations, do they behave in a worse way as a result of that?
I think we don't know this, to be honest. It's not well documented at the moment. I once went back to my MLH1 PMS2, let's say classic methylated cases, and actually, if you look carefully, a lot actually have very tiny bits of loss of MSH6. If you start to look carefully, I think we have missed that. So this triple loss is actually very common. And it can also be multifocal as well, and very tiny bits. So it can also something, I've seen cases where the MZ6 only a few glands, totally negative, and also multifocal. So I think we're underestimating how frequent this is. And I do not know, I mean, it could be sort of a surrogate or an indicator for tumor heterogeneity or even TMB in these cases. So whether it has an effect on prognosis, I don't know. I don't think it's been looked at.
Okay, interesting. And staying with the MSI group, you were describing a difference between the Lynch syndrome cases and the sporadic cases due to promoter hypermethylation of the MLH1 gene.
Yeah.
What's the explanation for why you get that kind of difference in behaviour, given that they're both acting through a similar mechanism of loss of mismatch repair?
Well, there's obviously there's some hypothesis on this, and also some additional work done, not necessarily by our group, but also is obviously. So one observation is that you see more intense lymphocytic infiltrate in the Lynch syndrome associated endometrial carcinomas. But the driving hypothesis could be that these patients I showed you these MMR deficient gland foci, and we see that if you look at the surgical resections for preventive hysterectomies, you see them a lot. So what I think is going on is in Lynch syndrome patients probably have lots and lots of latent precancers prior to the development of their cancer. So their immune system is probably very, very trained or has gone through sort of capturing these. So I think it could be a hypothesis that this is why they are the immune system probably is better at recognizing these tumors and responding to these cancers in the context of Lynch. I don't know if this is fully proven, but this is an hypothesis.
Yeah, there's been a similar hypothesis advanced for the equivalent situation in the colon and rectum, that in Lynch syndrome patients, the patient's immune system has been fully trained or primed essentially since birth. The immune system has had to deal with intestinal glands or endometrial glands that are defective for mismatch repair, are producing these frameshift peptides, and it primes the immune system in a much stronger way. So yeah, I think that sounds right. So going back to the Q&A box, there's a question from Marnix Janssen, who says, "Thanks, Tjalling, great talk. Your data show that molecular subtypes supersede morphologic subtypes (brackets sarcomas, clear cell, etc.). Is this correct? And do you disregard morphologic subtypes now during routine sign out? And if so, how does your management team respond?"
Very good question. Of course, this is this is what you get when you have a paradigm shift. You go from one classification to the other, and we're still used to the old one. We have all these data that the new one is better, but how do you go about this? So I basically report both. I don't ignore my histological subtype. I can easily say serous endometrial cancer, grade 3, MMR deficient, or polymutant, no problem. I also don't want to ignore it, because I still think that there might be information that we don't know yet. Prognosis is only one variable to look at. There can also be treatment response differences or other things that may be relevant. So I think I would still report it, but my clinical management team uses the most recent guidelines, and there you have in the table you have the classic system, and next to it when you have the molecular classification. And my team uses when you have the molecular classification. So they basically, in majority of the cases, the histological subtype is overruled and is in that sense disregarded.
Okay, thank you. I think we can squeeze in one last question from Kezia Gateskill. Thanks for a really great talk. I have a question about a double classifier case. Have you ever encountered a case which seems to be a typically p53 mutant serous carcinoma, but with focal MMR loss? If so, what, if any, significance do you think this might have?
Well, if you describe such a case, the first thing I would think is it maybe a POLE? Because the POLEs can have the nuclear atypia that looks like serous, and subclonality of MMR is often seen in POLE. So first exclude that, I'd say, and yeah, and then move further.
Okay, lovely. Thanks very much for that. And we've run out of time. We've just hit 10 a.m. So it just remains for me to thank Tjalling again for a really excellent talk with some fascinating data. Very enjoyable. Thank you very much for joining us.
Thank you. Bye-bye.
Thank you and bye.